I'm going to digress here just a little bit because we've done some significant tissues like loose connective tissue in which immune surveillance is one of the key features. We also have skin here, which is going to be dealing with the issue of immunology. Now, immunology is complicated, but I want you to have sort of a glancing sensibility of what's going on when we have an immune response. An immune response means ultimately that this particular cell here has, let me find my pointing tool. Okay, this particular cell here is a macrophage or a white blood cell, also known as a monocyte. It eats debris, including pathogens. And if it happens to eat an HIV virus, or if it happens to eat a COVID-19 SARS-2 virus, then it will display some aspect of that virus, usually its protein coat, on the surface. And we have what's here is an antigen, a foreign antigen. So I just need you to understand the macrophage eats it. It doesn't matter if the particle is alive or dead, right? So we can make vaccines with dead viruses or attenuated viruses or live viruses. In any case, whatever we use or whatever pathogen this monocyte or macrophage eats, it will display a portion of it out on its surface. And we have, how shall I say, lymphocytes, which can basically create receptors that can identify the antigen or bind to it, not necessarily identify it, but bind to it. And thus, we can mount an immune response. So, first of all, if the monocyte eats it and metabolizes it and displays it, it's called antigen presenting, then something called a T helper can recognize it. The T helper is at the center of this along with the monocyte or macrophage and activates two systems. There's a kind of system called cellular immunity and there's a kind of system called humoral immunity. Okay, and you might be more familiar with humoral immunity, in which a B cell gets activated and grows up to be a plasma cell, and we produce antibodies. So the idea here is that we're going to have antibodies, and as a result of the antibodies, we're going to be able to bind to the antigen, and it's usually going to be some portion of the virus. When we bind to it, then there are other systems that come into play and destroy that particular virus. Another aspect of this is that we create a special kind of T cell that goes out, and it's got a little receptor on its surface, and it seeks out these virally infected cells or cancer cells, tumor cells. In either case, once it identifies the cell, it throws a bunch of spears into the cell and it lyses the cell. Okay? So there's two different types of immune response. We'll get more into that in the second semester where we have a whole chapter that looks at this. But I need you to have sort of a glancing idea of what's going on, especially we're now in the midst of COVID-19 while I'm making this particular little video, and I want you to understand what is a vaccine. A vaccine is the ability through antibodies or through T cytotoxic cells to identify a pathogen that has infected a cell, or using this immunoglobulin or antibody that can bind to the pathogen and identify it that way. We have other systems like complement and so on that we'll be getting into that can destroy the cell once we've identified it. So I just need you to have a glancing idea that if the macrophage eats the pathogen, metabolizes it, presents it, identifies it to certain cells of the immune system, we can have an immune response. And that's the purpose of a vaccine, is to create an immune response that successfully deals with the disease. Okay, so as we look down through this lower portion down here, we can see that it's possible to have a viral infection. We end up with an illness as a result of it, and we produce an antibody. Eventually, the antibody helps us get rid of the infection. If we see that infection again, we get a little boost here, a little booster. These can actually be vaccines. We can get a vaccine without the illness and get an antibody. If it's not good enough in terms of concentration, we can get a booster shot from the vaccine and increase the antibody concentration in the blood. Okay, so that's the idea here. And I just wanted to present that right away so that I can go on to other things and talk about it. So you can imagine in loose connective tissue, wherever you see a macrophage, it's cruising around. Okay? And so we're at part 2 here now on page 152. And we have to decide, first of all, what cells are involved in the epidermis. Remember that the epidermis, let's go back for just a second here and look at our skin. The epidermis is this outer layer of the skin. Okay, it's the surface layer. It's made up of keratinocytes. And it is also the layer, if you go deep enough, where hair cells are produced. Also, this diagram doesn't show us, but it's also the place where fingernails and toenails are produced if you go deep to the cuticle or eponychium or whatever you want to, however you describe all of that. We'll get to that momentarily. But meanwhile, the cells of the epidermis. Okay, so we're going to be looking at the cells that actually produce the structure, which is the keratinocytes, and that's going to be the stratified squamous epithelial cells. And then we're going to have some other cells, melanocytes that make melanin, dendrite cells, which are those little macrophages that can cruise around and eat things and mount an immune response. We also have other cells that have to do with touch. Okay, so first of all, the keratinocytes are our first consideration here because they produce the structure of the epidermis. Keratinocytes. They're called keratinocytes because they produce a type of protein called keratin. The keratin is the protein that produces hair. It produces fingernails, and it also produces the topmost layer of the skin, keratin. Let's take a look over here at some skin. First of all, this is an actual photomicrograph, which is, once we learn what it's supposed to look like, then it'll become less boring. So let's come over here to the artistic rendition and see what's going on. We have actually, this is huge right through here. This is a callus right here. Do you see this stratum corneum? It's the dead layer of the skin. So we'll get to that in just a second. But first of all, here's the germ layer here. This is the basement membrane upon which the first set of cells exist. And these cells are basically replicating quickly. And we're going to be able to have daughter cells produced here. Now, these cells are held together by desmosomes, and so you'll notice that with the first layer of cells here, they're actually being held apart by the desmosome. Okay, so what do we call this? We call this the stratum spinosum, because they appear to have little spines on them, and by pushing the cells away from each other, this means that the capillaries in through here are able to diffuse nutrients out to these cells so that they get to eat waste products, get diffused back to the capillaries for removal from the system. Okay, what else do we have here? We have a neuron coming up this way, so we have a tactile. This particular cell, this actually receptor here is called a Merkel receptor, Merkel cell. It's hooked up to a little neuron. Over here we have a melanocyte, and we're producing melanin. Now, if you were to look carefully here, it's hard to make this out, but if you were to look at a real micrograph, these would be little particles of melanin, not just some continuous fluid or something like that. It would be like particles. Okay.